That brings us to our special senses. This is where it starts to get fun, because now you can see what in the hell is actually triggering all of these various different types of sensations. Okay, the ability to taste certain things. Fascinating. Very, very fascinating. The ability to understand why a taste sensation becomes so in your face, or why a smell sensation is so in your face, is based on the amount of receptors I'm stimulating, the amount of neurotransmitters that they release. Taste and smell are intimately connected to one another, right? These two sensations will affect each other. If you can't smell properly, food doesn't taste like it normally does. Okay? If your sense of taste is dulled, or if your sense of taste is altered at all, it can affect how you interpret smells. It's fascinating. It's so interesting. And some of the ion channels that are going to be involved here, like sodium is going to be involved here, shocking with some of these, okay? With COVID, it's affecting some of these receptors in the taste sensation realm. It's affecting the sodium channels associated with some of the ability to smell certain types of smells. We see the same thing with mucus production in the digestive tract, in the esophagus, really, with the mucociliary escalator. Again, it's sodium ion channels are going to be involved with that, or chloride ion channels are going to be involved with that. And COVID, that virus attacks those specific receptors. Same thing in the lungs. So these receptors that we're talking about with, hyperpolarization or depolarization a lot of times here, and involving various different mechanisms that we're going to be talking about, COVID attacks all of those receptors. And COVID causes events to occur even when they're not supposed to occur. And it can deaden some of these receptors, as it does when it comes to smell and taste. And we don't really understand if that's going to come back. Same thing, I just saw a report yesterday that looked at the eyeball, and how individuals who have had COVID are having some nodules develop on the back of their retina. We don't know what the hell that's going to do long term for these individuals. Nodules, they don't really exist back on the back of that retina. What does that mean for visual acuity? What does that mean for, long-term vision for that individual? We don't know. And this was just the first study I've seen involving COVID with the retina, and involving the development of nodules in there. The eyeball, for lack of, let me do one last left turn here. The eyeball has kind of like a boundary that prevents the immune system from getting there and mounting an immune response. So is it affecting those specific cells that are involved with that? I don't know. It's pretty interesting. So when it comes to this stuff, so our understanding of some of these things is just starting to come into fruition. They do a ton of research on taste and smell. If I get back on track here, they do a ton of research on these two. These things change dramatically over the course of your lifetime. Why? We don't fully understand. Sometimes taste and smells change dramatically with pregnancy. Hormones, right? And then sometimes they go back. Sometimes they don't. Sometimes it takes a few years. So they do a ton of research on these things. So even though we're going to talk about a bunch of stuff, in the grand scheme, when it comes to these two types of sensations, stuff may change in a handful of years. It is not uncommon when it comes to these two types of sensations here.
You did a lab like this in my anatomy class. It was really cool.
Mm-hmm.
So our teacher brought in a really big bag of Skittles,
Mm-hmm.
and she had us eat one regularly, and then she had us plug our nose and then eat one.
Mm-hmm.
And we were really surprised that we could still taste it, but it was very faint. It was kind of cool.
Very muted, yeah. It's crazy how they are so intertwined with one another. But it also goes with, if it smells bad, you're not going to eat it. And you're going to already have a negative association with it. And that's going to reinforce the concept that you don't want to try it. So, why does stuff have to, smell good? Because it influences your taste. And that goes to not only the taste sensations themselves, but your cognizant interpretation of it. So it's going to affect, that certain smells from growing up, when certain things smell bad, you got to stay away from it, right? Because that's what you're told. It's what you tell your kid when you're out running around and you see dog poop and they're like, I want to go touch it. They're like, oh, smelly. Oh, gosh, don't touch. Bad, bad. That already starting to hardwire them in. When it comes to taste sensations with children, that's why you do not introduce sweets first. You always introduce vegetables first, because sweet is going to be, it hardwires these taste receptors to be a hell of a lot more receptive to it. And so that's going to make them want those a hell of a lot more, as opposed to vegetables, which don't have that sweet sensation. And they're going to downplay some of the activity that should happen when you start to eat vegetables. And so they're going to hardwire them not to eat vegetables. So it's all based on these perceptions of taste and smell and how that happens. They are chemoreceptors. We mentioned that briefly earlier. These are doing both internal receptors and external, detecting chemical changes within the body. External receptors are going to be more of these type of chemoreceptors here, dissolved in food and drink, in air, and yes, I said they greatly influences one another. Here is where taste happens. Taste, the fancy name for taste is called gustation, and I just like saying it that way. The receptors for taste are found in your taste buds. And roughly it's around, I doubt I'll ask it, it's around 50 to 100 specialized epithelial cells, okay? And these are them right here, these gustatory taste cells right here, okay? That's them. They're just 100 roughly specialized epithelial cells. They have a much more elongated, microvilli that extend through this opening right there, and there's the opening. Okay, so it's an opening right there. And it's those little microvilli that kind of go out there and interact with the environment. Okay, is it normal to have hair on your tongue? Obviously not. Okay, I think there's a couple conditions associated with some hairs on the surface of your tongue. Ugh, there you go. That's I can't even think about that. I just can't. Okay, there's certain things I can do almost anything. I cannot deal with hair on the tongue, but that's just of recent. It just sounds disturbing. I don't like belly buttons. I don't like anything about a belly button. And eyeballs. Don't like eyeballs. Ugh, I just don't like them. Okay? So hairs on the tongue, that would definitely be one of them. But if I put my blinker on and get back in my lane, it's these microvilli that come up and interact with those chemicals that you have within the food. Obviously, that happens through chewing, saliva getting secreted, the chemicals getting dissolved in that saliva there and triggering those little taste receptors. We find all these little taste buds found in papillae, and we have three really major ones. They're all found in a specific region. I mean, again, that makes a good just general knowledge type of multiple choice type of question. But we have fungiform, circumvallate, and folate. And there you kind of say, this is all the anterior. There's your folate there on the sides. Okay. Here's your vallate, or this book calls them circumvallate, all right there. Okay. Located on the posterior aspect of the tongue. When it comes to taste sensation, it is all over the tongue. Okay? There's not like this is the sweet spot and this is the sour spot and this is the bitter spot. Okay? There's not generally one region like that. There is some debate as to when it comes to poisons and whatnot. They are mostly bitter in their taste sensation. You have a much greater concentration of bitter receptors found in the posterior aspect of the tongue. And that's just the body's last ditch effort to try to up and out something that can cause harm to the body. But other than that, bitter, sweet, sour, I mean, it's all over. Umami, it's all over the damn place. Okay. So there's not one specific locale. But we have fungiform, they're on the anterior. Facial nerve is generally what carries that information. Circumvallate is glossopharyngeal, and even the ones on the side are carried by glossopharyngeal. So circumvallate and folate, posterior 1/3, anterior 2/3, are going to be carrying this information. All right. A very generalized pathway here goes from either one of those nerves, anterior 2/3 or posterior 1/3. Okay, the facial or glossopharyngeal nerves. Travel back to the medulla oblongata. Right. It specifically synapses on the gustatory nucleus. I probably will not ask specifically what nucleus. Okay. Gustatory just means taste. It's just the taste nucleus. So, I mean, it makes sense that that would be the nuclei that it would go and synapse on because this is involving taste. But it's in the medulla oblongata. From the medulla, we go back into the thalamus. And then again, the thalamus is the great distributor of information. All right. Everything pretty much goes through the thalamus there. It determines what goes up into the upper portions of the brain in the cerebrum there to be interpreted ultimately. All right. So it's going to filter out a lot of stuff. Then we go to the primary gustatory cortex, insula, somatosensory cortex, and the parietal lobe, prefrontal cortex. Again, this is all how we interpret what we are tasting. Taste sensation. Okay, sweet, sour, salty, bitter, umami, all those taste in that insula. The rest of it is just, somatosensory cortex allows us to interpret that taste, okay, or that texture, that touch. And then the prefrontal cortex, again, we're just, that's where we learn what we really like or we don't like. So all of those regions are tied into this tongue of ours. As any other cell does, it's going to depolarize. Okay? It's going to release neurotransmitters. Okay? We'll get into those here. Glutamate is one of them. Again, the microvilli, they're in those taste cells, come in contact with those chemicals that help us to interpret these things. Each taste bud has taste cells sensitive to each category of taste. And again, this is something which seems pretty obvious, but this is something relatively new within the last several years, 10 years or so. Each taste bud has a taste cell sensitive to every category of taste. And these are your 5 categories. Salty, sour, sweet, umami, and bitter. Temperature, texture, all of this adds into how we taste. Especially temperature with capsaicin receptors and whatnot. Okay. But if you've ever eaten something warm and compared it to something cold, it tastes different, right? Pizza tastes a hell of a lot different when it's cold out of the fridge first thing in the morning than when you got it the night before when it was piping hot, right? So temperature influences this. But when it comes to these taste sensations, here they are. And I believe I kind of have them all listed on the next slide. Okay. But there you can see salty and sour. These are basically ionic trophic. I have a specific ion that is binding to that microvilli there, that villi, that little hair cell, leading to a depolarization event. As you can kind of see, salt responds to sodium. NaCl, salt, right? Sour responds to hydrogen ions. And these are going to be found within the foods. As you chew them up, mash it up, mixes with the saliva, these ions, these chemicals, right? They get in there, bind to that little hair, and cause a depolarization event. When the cell depolarizes, calcium channels open up. And that helps us to stimulate the release of even more neurotransmitters, just like we saw when we saw what happened at the synaptic node, right? As the action potential got closer to the synaptic knob, voltage-gated calcium channels opened up, calcium rushed in, and released the neurotransmitter acetylcholine. I'm not getting into all the different types of neurotransmitters here, right? But neurotransmitters get released, and that a depolarization event travels down that sensory nerve into the brain, and we interpret that as salty. The more sodium there is, the more receptors I can trigger and open up, and the more calcium I can come in, and the hell of a lot more neurotransmitters I can release, and that increases the frequency of action potentials. Same thing with sour. The more hydrogen ions I have in it, the more depolarization occurs within the cell. The more calcium voltage-gated channels open up, the more calcium comes in, the more neurotransmitters I release, yada, yada, yada. It's just like every other thing we've talked about, it seems like. Again, sodium and hydrogen ions. What triggers the depolarization? It's those two ions. Bitter, sweet, and umami are all G protein coupled reactions, G protein coupled receptors. It triggers a secondary messenger system. Glucose. Or glutamate binds to receptors. Okay. Sugars, glutamate, glucose. Okay. Binds to receptor. That triggers a G protein coupled receptor, a secondary messenger system. Okay. It activates a number of gustasins. Okay. This is just a general name for the secondary messenger inside a taste receptor. Gustation is taste. Gustasins is just that enzyme. Okay. Here's that gustasin that I've now stimulated to be active. It goes here and actually closes potassium channels. Potassium was associated with a hyperpolarization. So if that closes, that triggers the release of more neurotransmitters because now this thing can be depolarized. So the more glucose, let's just say, that I shovel in my mouth, the more I can open up and bind to it, the more I can open up these G protein coupled receptors, the more gustasin I produce, the greater these potassium channels stay closed, and the more neurotransmitters I release. And I interpret that as sweet. If I add a hell of a lot of sugar there, then I can interpret that as really, really sweet.
What causes that like excessive sour, like taste on your tongue that it feels different if you accumulate a lot of that?
It's the hydrogen ions that are within it that give us that sour taste sensation. So it's the hydrogen ions that are going to be binding with those receptors here on those little hairs. And the greater number of hydrogen ions, I interpret that as sour. Okay. When it comes to bitter, quinine is going to be found within is the chemical found within stuff that's bitter. And again, we are triggering a secondary messenger system, a gustation. This will trigger calcium to be released from the endoplasmic reticulum. Remember, endoplasmic reticulum, it can be specialized to store calcium. And here it's specialized to store calcium. That pumps out calcium, just like it does in skeletal muscle tissue, right? Sarcoplasmic reticulum, endoplasmic reticulum, right? Endoplasmic reticulum gets released from that, calcium gets released from that endoplasmic reticulum. And again, calcium leads to depolarization. And so I send more action potentials. More quinine I have bound to all of these receptors here. The greater the amount of calcium gets released, the greater number of vesicles get fused with the plasma membrane here, dump the neurotransmitter, exocytose it, diffuse across the synapse there, and bind to the receptors there on the sensory neuron. This one's very sensitive. Okay. Bitter is very sensitive. Again, bitter is more associated with poisons, stuff that can harm the body. So when it comes to this one, it takes very little bitter to produce a very in-your-face taste. There are a number of other molecules as well, guys. I just kept it really simple: sodium, hydrogen, sugar, quinine, glutamate when it comes to umami. Okay. But there's inositol, inositol triphosphate. There's a thing called DAG, which is diacylglycerol. Cyclic AMP is involved with this process in some of these as well. So there is a little bit more, but these are the major chemicals that trigger these taste sensations. Sodium. Sodium binds to the receptors on those hairs. Depolarization. Sour the same way. These two are ionotrophic. Okay. These three are basically metabotropic, where they trigger secondary messenger systems. And it's just gustation, all right? That's our general G protein here. So sugar for sweet, glutamate for umami. It binds those receptors, kicks off the secondary messenger system, and for those two, it actually closes off potassium channels. Cell becomes, instead of hyperpolarized, depolarized. And now I can send the electrical action potential, and I can interpret that as sweet or umami, depending upon is it sugar or is it glutamate binding to it. Bitter, quinine is the chemical that binds to the receptor here, activates the gustation to cause the endoplasmic reticulum to release calcium. Calcium causes the vesicle to move towards the membrane, exocytose, diffuse, send the action potential to the brain, to which I interpret as bitter. I talked about hardwiring there. So if child, baby, whatever, infant, consumes something that is excessively sweet, you can see how it'll hardwire those taste receptors, because now it's going to be all right. So that's what sweet is. And if you're flooding that mouth of that little one with a very sweet product and a lot of sweet product, it can really trigger those receptors to be like, okay, I need this because woo, that tasted fantastic. And if you give them something excessively sweet when they first can start consuming food, it can hardwire those things. By not introducing sugar so early as a first food, that it's rice cereal, ugh, bland, right? Vegetables, right? They don't have a very sweet taste. So you don't hardwire those taste receptors to be like, okay, I need to be flooded with this much sugar to get the same sweet. Because then you just keep adding more sugar and sugar to stuff, and then that's not conducive for health.